And there's a sleeping sickness in people and Nagana in animals, and clearly the animal disease is much more widely distributed than the human disease. Do the animal infective parasites infect people?
No. So the animal infective parasites are killed by things in our blood, and that's why we don't get infected by them. But there are parasites that will infect humans.
So why would it be important to identify those components in human blood that protect us against infection with the animal infective trypanosomes?
Well, if we knew what they were, perhaps we could give them to the animals, and that way we could cure them and it would effect as a drug. The other reason is, if we knew what they were, could we give humans more of them and potentially be able to kill the human infective parasites? That's something we don't know.
And how far have you gotten in your research to identifying these factors?
Well, we know that they're what we call the good cholesterol. So there's good and bad cholesterol in our bodies, and as it turns out, the factors that are able to kill cattle infected parasites are the good cholesterol. And the protein components that we're aware of at the moment are two. One is called apolipoprotein L1, and that has been shown very nicely that it will form pores in the membranes of susceptible trypanosomes. The other protein is called haptoglobin-related protein, and it's in every single trypanosome lytic factor that has been studied to date, in every single good cholesterol. And we don't know what it does or why it's there, but we believe that it performs some function to help these factors work to their maximal capacity.
Given that the active ingredients are proteins and therefore reasonably can be expressed as genes, do you envisage using the genes encoding these proteins to modify a susceptibility of animals to animal infective trypanosomes?
That would be a dream. I mean, it's been shown very nicely that the apolipoprotein L1 protein, if you put that into an animal, it is very effective at killing parasites. And if we could give that to cattle as a gene so that they would naturally make it just like humans do, rather than having to give them the drug every time they got the disease, it would be far more effective, and we would have truly trypano-resistant cattle as opposed to trypano-tolerant.
So you're expressing the idea that you would modify the capability of the animal to resist infection by a germline alteration. It would now transfer with sperm and egg.
That would be correct, yeah.
And what kind of technology is used to achieve that?
Unfortunately, it's taking a gene from a human and then cloning it into the cells of a cow, and then giving those, if you can generate sperm that contains this gene, then you can use a male animal to fertilize all the female animals, and in that way you can transfer the gene to the progeny. The way people do it currently in mouse genetics is they give the gene to the mother, and the mother then has the offspring. It would be far more effective to put the gene into the male line and have the male transfer it.
So up to this point, we have focused our discussion in the context of modifying animals to resist animal infective trypanosomes. What has been learned in the context of your study of how human serum kills the animal infective trypanosomes to tell one how one might modify human serum to now kill human infective trypanosomes?
So some fabulous work has been done by a group in Belgium led by Etienne Pays, and he discovered the lipoprotein L. And what that does is it forms pores under acidic conditions in the membranes of the parasite, and the parasite dies as a consequence of that. So if we could better direct pore-forming molecules to trypanosomes, it would be a fabulous way of killing all species, because currently there is the West African Trypanosomosis, Trypanosoma gambiense, which is completely resistant to our trypanosome lytic factors, and it's completely resistant to the apolipoprotein L. And so we currently have no way of killing the human infective parasites in West Africa. However, there's been some fabulous work done by Terry Pearson, and he's shown that microbicidal peptides, which are again very tiny peptides that form pores in membranes, are able to kill selectively trypanosomes. And so if we could attach these microbicidal peptides to something that would target specifically to the trypanosome, then we would get a very effective killing molecule. And there's a guy who's going to present work tomorrow who is named Patrick de Basilier, and he's made these wonderful nanobodies where you take a very small camel antibody and you add the lytic factor to it, and the antibody is specifically directed to the trypanosome. So I think this is a fabulous technology, and if people won't accept the genetically modified cattle, then perhaps they would accept this protein therapeutic as a way of curing trypanosomosis.
And that then leads me to ask you, is there an ethical or moral difference between having a constitutive production of a protein moiety that will kill parasites, for example, as opposed to packaging it in a bottle and injecting it?
It would be much better to have the animal make this little nanobody with the nasty toxin attached to it, because it would be self, and you would never have to worry about raising an immune reaction against that protein and clearing it from your system. And the problem with repeatedly injecting protein to cure yourself of a disease is eventually you'll recognize it as foreign, you'll make antibodies to it, and you'll completely clear it. And so, again, we're back to the genetically modified cow, and it would be really the best way to go.
So now you would be creating a situation where you'd place the parasite under a selective pressure from molecules either that you've constructed artificially or which are natural antimicrobicidal compounds. Do you think there's a danger that you might select for novel strains of virulent parasites?
There's always that danger when you apply drug pressure to any pathogen, and we've seen this with antibiotic resistance in bacteria, and people are now scrambling to try and deal with that and come up with novel therapeutics. But it would buy a lot of time for a lot of people, and perhaps get people out of the cycle of poverty and not having enough food and all their cows being sick, not having enough milk, and just give people a chance to move on and do different things in this world instead of constantly trying to take care of their sick animals and their sick cells.
And do you think because of the combination of microbicidal peptides and the fragments of the human lytic serum lipoprotein materials, together with potential targeting devices in the parasite, that one might make animals bearing multiple resistance markers?
Wouldn't that be great? Yes. What's happening with AIDS research now is people say you shouldn't use just one line of drug therapy because the virus can really easily mutate and become resistant to that. And what should be used in all therapies against infectious agents is multiple targets, because then you're requiring the pathogen to make multiple mutations to resist all of the different assaults. And so it's far better to have many different ways of killing something given to one person than to just take one drug.
Thank you, Jane. Thank you very much.